Hello class, my name is Lindsay Pappas, and if I haven't met you yet, I am a first year PhD student in the Department of Nutrition Sciences here at UAB. So my topic today is called The Effect of Exercise and Protein Source on Characteristics of Metabolic Syndrome in Obese Rats. And this is actually the first study that I was involved in from conception to completion, so to speak, back in 2014 as an undergraduate. My mentors, research assistants, and I were given the opportunity to design and carry out an experiment with extra offspring that he happened to have, so this is what we came up with. So just a brief overview of metabolic syndrome. It's a cluster of the conditions on the left-hand side, which are decreased HDL cholesterol, the presence of insulin resistance, increased blood pressure or hypertension, abdominal obesity, as well as elevated triglycerides. And having 3 or more of these conditions, so to speak, is criteria for metabolic syndrome. And I should also mention that these are thankfully modifiable risk factors, so to speak, but the presence of all of these, or even just a couple of them, increases one's risk for developing cardiovascular disease as well as type 2 diabetes. So a recent statistic that I found was that approximately 30 to 40% of people by the age of 65 will actually develop metabolic syndrome, which I thought was quite shocking. And of course, the main driver is weight gain. Thus, no surprise that having obesity increases one's risk of developing the characteristics that I mentioned of metabolic syndrome. And of course, with obesity, exercise and diet is really critical for both obesity as well as metabolic syndrome. And the current recommendations to decrease the conditions or one's risk for developing metabolic syndrome as a whole is to reduce total weight by approximately 5 to 10%. All right, so moving on to proteins. We know from our class this semester that dietary proteins have unique metabolic and physiologic properties, and because of this, they have been utilized widely in dietary interventions in both preventative and therapeutic approaches. Proteins support lean mass by maintaining muscle mass, and they also stimulate protein synthesis. And of course, high protein diets have been used extensively to promote weight loss, as reported by the literature. More recently, there's growing evidence that suggests that not only the quantity of proteins, but also the source of the proteins could be a determining factor on their overall effect on food intake, body weight, body composition, and glucose metabolism. Whey and soy proteins are 2 major sources of proteins in the Western diet today, and they are considered high quality proteins, and that's because of their high biological values. They're also categorized as fast proteins, and this is because they are quickly digested and absorbed compared with slower proteins like casein. And the favorable effects of whey and soy on regulation of food intake, body weight, and glucose metabolism have been previously reported by my mentor back in undergrad, but the effect in combination with exercise was unknown prior to this experiment. Moving forward, physical activity combined with a balanced diet can reverse the effects and reduce the risk of metabolic syndrome. However, the role of the source of protein in combination with physical exercise on symptoms of metabolic syndrome and the treatment of obesity was unclear. So the objective of the study was to evaluate the effect of whey protein and soy protein-based diets with and without exercise in obese female Wistar rats. And our hypothesis was that dietary protein source influences the effect of exercise on food intake, body weight, and body composition in obese rats. All right, so moving on to the experimental design, we induced obesity in 48 post-weaned female Wistar rats. And you can see the composition of their diet on the right hand in the table. What this was, if I can recall correctly, it was a combination of sugar, a standard protein source, as well as evaporated milk, and they absolutely loved it. They could not get enough of it. And unfortunately, because it was all sugar, it was really, really sticky and messy. But anyways, so after 12 weeks, we allocated them into 4 groups, 12 rats per group. One group received the whey protein plus exercise, the other soy protein diet plus exercise, and then, of course, whey and soy without exercise. And the exercise intervention was 30 minutes on the treadmill 3 times per week, and let me tell you how hard it was to run rats on treadmills when they are extremely obese. But thankfully, towards the end of the study, they started to lose a significant amount of weight. Continuing with the study design, we measured the animal's body weight and food intake at least 3 times per week. And then at week 20, we also did some additional assessments, which included systolic and diastolic blood pressure, and short-term food intake at 1, 2, and 12 hours in response to both whey and soy protein preloads. And at this time, an oral glucose tolerance test was also conducted. At the end of the study, and after 12 hours of fasting, the rats were euthanized, and we drew samples of blood to test for plasma concentrations of glucose, insulin, and the food intake regulatory hormones, peptide YY or PYY, and ghrelin. The effect of the diet and exercise and their interactions on body weight, food intake, systolic and diastolic blood pressure, oral glucose tolerance test, and HOMA-IR, and plasma glucose, insulin, PYY, and ghrelin were analyzed by 2A ANOVA. When repeated measures were made over time on body weight and food intake, the PROC mixed model procedure was used with diets, exercise, and time as the main factors. When these interactions were statistically significant, a one-way ANOVA followed by a post-hoc Tukey's test was conducted to evaluate the treatment effects. And of course, all analysis were conducted using SAS, and significance was defined as having a p-value of less than 0.05. Now on to the results. Let's first start with body weight. So, as I mentioned, we fed the rats an obesogenic diet for 12 weeks. As you can see, each of the 4 groups was pretty similar in body weight at the beginning of the actual study, between 400 and 420 grams for about an average of 410 grams, which is actually very huge for a female rat. And if you take a look at the key, the triangle denotes soy protein, no exercise. The square is whey protein, no exercise. The asterisk is soy protein, exercise. And then the diamond is whey protein, exercise. So of course, just by looking at this, you can tell that body weight was lower in all groups at week 20 compared with baseline, which is week 12. Both body weight and body composition were affected by the source of protein in a significant manner, but exercise was not. And as you can see, whey protein resulted in lower body weight compared with all other groups at week 20. Now, taking a look at body composition, as before I mentioned, body weight was lower in the whey protein no exercise group compared with the other groups at week 20, and body weight was mainly affected by the source of protein. In this case, body composition was only affected by exercise only. The whey protein exercise group showed lower abdominal fat and higher lean mass, which is represented as percent body weight, compared with baseline, and also compared with the other intervention groups at week 20. Now, taking a look at food intake over the course of the study, we can see that regardless of the protein source, long-term food intake was significantly higher in those treatment groups who exercised compared with the non-exercise groups. So the highest intake in food was the soy protein with exercise, followed by the whey protein with exercise, and then of course the soy protein no exercise, and finally the smallest amount of food intake was by the whey protein no exercise. Looking at short-term food intake, this was measured post-preload gavaging of the animals based off of their diet, so either soy or whey protein preload. And then food intake was measured at 1, 2, and 12 hours. And there was no effect found on protein and or exercise on short-term food intake, but food intake was higher in the exercise group compared with the no exercise group within the first hour of food intake. In conclusion, and just to recap, regardless of the source of protein, long-term food intake was significantly higher in exercise groups compared with non-exercise groups. Body weight was relatively lower in all groups compared with baseline at week 20. However, body weight was lower in the whey protein no exercise group compared with all other groups at week 20, which signifies that body weight was affected by the source of protein. On the other hand, body composition was affected by exercise only. The whey protein exercise group had lower abdominal fat and higher lean mass compared with baseline, and also compared with other groups at week 20. And I didn't present the following data to you just for the sake of time, but I'll go ahead and outline it. So, consistent with the higher food intake observed in the exercise groups, their fasting plasma concentration of ghrelin was also higher compared with the non-exercise groups, and also compared with baseline. There was no effect of either exercise or diet on fasting plasma concentrations of glucose, insulin, and insulin-glucose ratio at week 20. Additionally, diastolic blood pressure and pulse were influenced by diet at week 20. Diastolic blood pressure was significantly higher and pulse was significantly lower in the whey protein groups compared with the soy protein groups. Pulse was also lower in the whey protein groups compared with baseline at week 20. So the results of this study indicate that both the source of protein as well as exercise may be dominant factors in the treatment of obesity in obese female Wistar rats. While exercise influences food intake, the protein source determines the effects of exercise on body weight and body composition with whey protein showing a more favorable effect. All right, let's take a look and kind of uncover why whey protein had a more favorable effect as a source of protein compared to soy protein. There are differences in the amino acid composition as well as differences in their post-absorptive metabolism, which may explain why whey protein was more successful in the study. Whey protein does have a higher concentration of branched chain amino acids, which has been shown to independently stimulate muscle protein synthesis compared to soy. On the other hand, soy has been shown to result in less protein synthesis, and this is because a larger portion of amino acids are degraded into urea after the ingestion of soy protein compared to whey protein. As for further directions for this area of research, definitely a further look into the exercise component in terms of duration and intensity, just because this study was only an 8-week long intervention of mild to moderate exercise. So it would be interesting to see something more intense for a longer duration. Finally, I'd just like to quickly acknowledge my previous mentor, Dr. Jahan Mahan, as well as Dr. McGarry, and the two research assistants that I worked closely with throughout my undergrad years, Shauna and Val. And that's Shauna in the picture with the rat treadmill. If you've never seen one, it is pretty much what you would think it would look like. But yeah, thank you so much, and I'm more than happy to answer any questions you have.